Which bacteria were used to clean the beach after the Exxon Valdez oil spill?
B. Bacteria from the genus Pseudomonas are capable of degrading oil as their source of nutrition. This microorganism was used to clean up the spill that emerged from the Exxon Valdez. In order to improve the efficiency of this bacteria, nitrogen and phosphorus were introduced to beaches polluted by the spill. The number of oil-degrading bacteria on the beaches increased sufficiently enough to remove much of the oil from the beaches.
What is the function of chloroplasts?
Chloroplasts are the structural and functional units where photosynthesis takes place, the process whereby green plants use light energy for the synthesis of organic molecules from carbon dioxide and water, with oxygen released as a byproduct. They contain the green pigment chlorophyll, which traps light energy for photosynthesis.
What is torpor?
Torpor is a short-term decrease in body temperature and metabolic rate. Animals such as hummingbirds and bats go through daily periods of torpor that allow them to reduce their energy requirements at night or when hunting is poor. Torpor can be considered as a type of brief sleep, but it is distinct from a state from hibernation.
Can animals recognize different languages?
Scientists have compared language discrimination in human newborns and cotton-top tamarin monkeys. Each group was presented with 20 sentences in Japanese and 20 sentences in Dutch. Infant reactions were gauged by their interest in sucking on a pacifier. When infants first heard sentences in Dutch, they sucked rapidly on their pacifiers. But after a while they grew bored with the Dutch sentences, and the rate of sucking slowed. When someone started speaking in Japanese, they showed increased interest by increasing their rate of sucking. Language discrimination was studied in tamarin monkeys by changes in the facial orientation toward or away from the loudspeaker. Similar to the infant reactions, the monkeys looked at a loudspeaker broadcasting Dutch sentences and looked away when bored. When someone started speaking Japanese sentences, they looked back at the loudspeaker. Results indicate shared sensitivities between monkeys and humans in the ability to discriminate between languages.
What mushroom has been identified as a fossil?
Mushrooms are rarely found in fossil records since there is little in the structure of a mushroom that can be fossilized. In 1992, researchers in the Dominican Republic discovered a fossil of a fleshy, gilled mushroom. The mushroom, Copernites dominicana, is believed to be 35 million to 40 million years old and is the only known fossil mushroom from the tropics. Microscopic examination suggests it is related to modern-day inky cap mushrooms.
Why do ecosystems need decomposers?
While energy flows through ecosystems in only one direction, entering at the producer level and exiting as heat and the transfer of energy as biomass, to consumers, chemical compounds can be reused over and over again. In a well-functioning ecosystem, some organisms make their living, their niche, by breaking down structures and recycling the compounds. These organisms are known as decomposers. Without these organisms, the chemicals used to build a tree would remain locked in the tree biomass for eternity instead of being returned to the soil after the tree's death. From this soil will spring new growth, beginning the cycle once again.
What is the Kelvin temperature scale?
Temperature is the level of heat in a gas, liquid, or solid. The freezing and boiling points of water are used as standard reference levels in both the metric centigrade and the English system, Fahrenheit. In the metric system, the difference between freezing and boiling is divided into 100 equal intervals called degree centigrade, degree C. In the English system, the intervals are divided into 180 units, with one unit called degree Fahrenheit, degree F. But temperature can be measured from absolute zero, no heat, no motion. This principle defines thermodynamic temperature and establishes a method to measure it upward. This scale of temperature is called the Kelvin temperature scale, after its inventor, William Thomson, Lord Kelvin. 1824 to 1907, who devised it in 1848. The Kelvin, symbol K, has the same magnitude as the degree centigrade. The difference between freezing and boiling water is 100 degrees, but the two temperatures differ by 273.15 degrees. Absolute zero, which is minus 273.15 degrees Celsius on the centigrade scale.
What predicts whether one animal will help another?
Usually animals that help one another are of the same species, conspecific, and are most likely to be genetically related. The closer the relationship, the more likely helping will occur. This is demonstrated among vampire bats, where bats returning to the roost may share blood with those who have not fed. Most sharing occurs between close relatives, e.g. mother and offspring. However, unrelated but closely associated individuals may exhibit reciprocal altruism, where help is given to those individuals most likely to return the favor.
Skinner related to operant conditioning?
B. F. Skinner, 1904 to 1990, was an American psychologist who extensively studied trial and error learning in animals, later known as operant conditioning. A standard setup for his research involved the following: an animal is placed in a cage, known as a Skinner box, that has a bar or pedal that yields a food reward when pressed. Once the animal has practiced the behavior, it will continue to press the bar repeatedly. Having learned to associate this activity with food by releasing food only when the animal completes some task, the observer can train the subject to perform complex behaviors on demand. These operant conditioning techniques have been used to teach behaviors such as training pigeons to play table tennis with their beaks.
What is pharmacogenomics?
Pharmacogenomics is the use of DNA technology to develop new drugs and optimize current drug treatment to individual patients. For example, the interaction of a drug with a specific protein can be studied and then compared to a cell in which a genetic mutation has inactivated that protein. Its potential is to tailor drug therapy to an individual's genome. A tailoring that could reduce adverse drug reactions and increase the efficacy of drug treatment.
How do schooling fishes swim so close without colliding?
A fish school travels in a smooth movement. Each fish responds to movements of other fish, such that if one fish changes direction, all of the others will move accordingly. With eyes placed on the side of the head, fishes can see what is next to them and use this to move. A complex combination of hearing, lateral line system, sight, and smell allows fishes to establish position and direction in a school.
What is a race?
The term race was originally used to describe subspecies. However, as genetic analysis of humans has shown, there is greater genetic variation within geographic subpopulations, races, than among the human population as a whole. In other words, there is so much genetic overlap between groups formerly designated as races that the term is meaningless and biologically indefensible.
What is a life history table?
A life history table, also referred to as a life table, is a table that shows both survival and death rates in a specific population or organisms. The life table is patterned after actuarial tables used by insurance companies.
What is a bar body?
In 1949 Dr. Murray Barr, 1908 to 1995, noticed a dark body in the neurons of female cats. It was later identified as a structure found only in the nucleus of females. It was named a Barr body in honor of its discoverer.
How are cell structures divided during meiosis?
Meiosis is just part of the larger process of gametogenic production that occurs in either the ovaries or testes. During sperm formation, spermatogenesis, the cells that eventually become mature sperm go through two successive meiotic divisions. This results in 4 haploid spermatids that will develop into mature sperm cells. The sperm are then reconfigured into a cell that is specialized for one thing, fertilizing an egg. The mature sperm is basically a nucleus with one set of chromosomes, mitochondria, and flagellum for propulsion. In humans, the cells that will develop into mature eggs are present in the ovary before 60. The oogenesis, egg formation, ever occurs. Immature eggs, oogonia, remain in a stage referred to as meiosis I until they mature during puberty. During the stage of meiosis II, oocytes are ready to be released, but will not fully complete the meiotic process until after fertilization takes place. During the progression from one diploid oocyte to 4 haploid cells, cytoplasm is shunted unequally to only one cell. The end result is one large mature ovum and 2 or 3 very small haploid cells called polar bodies.
What is the relationship between metabolism and lifespan?
Several studies have concluded that a program maintaining proportions of carbohydrates, lipids, and proteins, but reducing the consumption of calories, can increase longevity. However, these conclusions have only been demonstrated in tropical guppy fish and the worm Sinorhabditis elegans. To date, there have been no large clinical studies that have demonstrated the value of this regimen for humans.
How do aseptic procedures prevent contamination?
The goal of aseptic procedures is to keep the organisms used in an experiment separate from the millions of other organisms in the environment. These procedures focus on the way to transfer organisms from test tube to test tube, from test tube to flask or petri dish, and from petri dish to petri dish or flask.
How do continuous feeders differ from discontinuous feeders?
Continuous feeders, also known as filter feeders, are aquatic animals that constantly feed by having water filled with food particles, e.g., small plankton or fishes, entering through the mouth. Continuous feeders do not need a storage area, such as a stomach, for food. Discontinuous feeders must hunt for food on a regular basis. They need a storage area to house food until it is digested.
Who discovered how muscles work?
Hugh Huxley, 1924, and Andrew Huxley, 1917, the scientists were unrelated. Researched theories regarding muscle contraction. Hugh Huxley was initially a nuclear physicist who entered the field of biology at the end of World War II. He used both X-ray diffraction and electron microscopy to study muscle contraction. Andrew Huxley was a muscle biochemist who obtained data similar to Hugh's, indicating that the contractile proteins thought to be present in muscles are not contractile at all, but rather slide past each other to shorten a muscle. This theory is called the sliding filament theory of muscle contraction.
How has centrifugation been used in biological applications?
Biologists primarily use centrifugation to isolate and determine the biological properties and functions of subcellular organelles and large molecules. They study the effects of centrifugal forces on cells, developing embryos, and protozoa. These techniques have allowed scientists to determine certain properties about cells, including surface tension, relative viscosity of the cytoplasm, and the spatial and functional interrelationship of cell organelles when redistributed in intact cells.
What methods of analysis are used for DNA profiling?
Both RFLP, restriction fragment length polymorphism, and PCR, polymerase chain reaction, techniques can be used for DNA profiling. The success of either method depends on identifying where the DNA of two individuals varies the most and how this variation can be used to discriminate between two different DNA samples. Since RFLP uses markers in regions that are highly variable, it is unlikely that the DNA of two unrelated individuals will be identical. However, this method requires at least 20 nanograms of purified, intact DNA. PCR-based DNA fingerprinting is a rapid, less expensive method and requires only a very small amount of DNA. As little as 50 white blood cells.
Where do scleriids occur in plants?
Scleriids occur in the roots, stems, and seed coats of plants. Groups of scleriids in the soft flesh give pears their characteristic gritty texture. Scleriids also give nutshells and seed coats their hardness.
What is the Hardy-Weinberg theorem?
The Hardy-Weinberg theorem, formulated in 1909, extends Mendel's laws to natural 392 populations. It explains in a formula what happens to the allelic end. Genotypic frequencies as alleles are passed between generations. The Hardy-Weinberg equilibrium is based on the following assumptions. 1. The population size is large. 2. Individuals mate randomly within the population. 3. There are no mutations. 4. There is no input of new alleles from outside sources, and 5. There is no natural selection. Such that each new allelic combination has equal chances of survival.
What animals are members of the phylum Cnidaria?
Corals, jellyfishes, sea anemones, and hydras are members of the phylum Cnidaria. The name Cnidaria, from the Greek term cnide, meaning nettle, and Latin term aria, meaning like or connected with, refers to the stinging structures that are characteristic of some of these animals. These organisms have a digestive cavity with only one opening to the outside. This opening is surrounded by a ring of tentacles used to capture food and defend against predators. Cells in the tentacles and outer body surface contain stinging, harpoon-like structures called nematocysts. Cnidarians are the first group in the animal hierarchy to have their cells organized into tissues.
What is anagenesis?
When a species gradually changes over time to the extent that it becomes a new species but does not give rise to additional species, no divergence, this is described as anagenesis.
What is the chemical composition of chitin?
Chitin, found in the exoskeletons of insects and other arthropods, is a glucosamine polysaccharide with the formula of C30H50O19N4 and a molecular weight of 770.42. The basic units of this substance are linked together by condensation reactions to make up long chains. Hydrogen bonds link the chains together and help make chitin rigid and strong. It is a white, amorphous, semi-transparent mass that is insoluble in common solvents like water and alcohol.
How did geology influence Darwin?
While traveling aboard the HMS Beagle, Charles Darwin. Read The Principles of Geology by Charles Lyell, 1797–1875. Catastrophism was the popular theory of the time about the forces driving geological change. Lyell's theory suggested that geologic change was not solely the result of random catastrophes. Rather, he proposed that geologic formations were most often the result of everyday occurrences like storms, waves, volcanic eruptions, and earthquakes that could be observed within an individual lifetime. This idea, that the same geologic processes at work today were also present during our evolutionary past, is known as uniformitarian. This conclusion also led Lyell and, before him, James Hutton, 1726–1797, to suggest that Earth must be much older than the previously accepted age of 6,000 years. Because these uniform processes would have required many millions of years to generate the structures he observed. Reading Lyell's work gave Darwin a new perspective as he traveled through South America and sought a mechanism by which he could explain his thoughts on evolution.
What is the Innocence Project?
The Innocence Project is a public law clinic at the Benjamin Cardozo Law School in New York that uses biotechnology, specifically DNA evidence, to reopen cases of people who have been wrongly convicted of crimes.
What were the first vascular plants?
The word vascular comes from the Latin word vasculum, meaning vessel or duct. It is believed that the first vascular plants were members of the division Rheniophyta, which flourished about 400 million years ago but are now extinct. Members of the extinct genus Cooksonia, named for the paleobotanist Isabel Cookson. 1893–1973, were the first vascular plants to be identified. These plants were only a few centimeters tall, with water-conducting cells present in their stems but without roots and leaves.
What are bioluminescent bacteria?
Bioluminescence is the production of light, with very little heat, by some organisms. The light-emitting substance, luciferin, in most species is an organic molecule that emits light when it is oxidized by molecular oxygen in the presence of an enzyme, luciferase. The enzyme picks up electrons from flavoproteins in the electron transport chain and then emits some of the electron's energy as a photon of light. Bioluminescence is primarily a marine phenomenon occurring in many regions of oceans or seas. One example is the Milky Sea found in the Indian Ocean, where the sea appears to have a soft, white glow.
What are the main functions of the plasma membrane?
The main purpose of the plasma membrane is to provide a barrier that keeps cellular components inside the cell while simultaneously keeping unwanted substances from entering the cell. The membrane allows essential nutrients to be transported into the cell and aids in the removal of waste products from the cell. The specific functions of a membrane depend on the kinds of phospholipids and proteins present in the plasma membrane.
Which criteria were used to form what is referred to as the classical approach of bacterial taxonomy?
In this approach, bacteria are grouped into genera and species on the basis of the following characteristics. 1. Structural and morphological characteristics including shape, size, arrangement, capsules, flagella, endospores, and gram stain. 2. Biochemical and physiological traits such as optimum temperature and pH ranges for growth, oxygen requirements, growth factor requirements, respiration, and the fermentation of end products. Antibiotic sensitivities, and types of carbohydrates used as an energy source.
What is genetic engineering?
Genetic engineering, also popularly known as molecular cloning or gene cloning, is the artificial recombination of nucleic acid molecules in a test tube, their insertion into a virus, bacterial plasmid, or other vector system, and the subsequent incorporation of the chimeric molecules into a host organism in which they are capable of continued propagation. The construction of such molecules has also been termed gene manipulation because it usually involves the production of novel genetic combinations by biochemical means. Genetic engineering techniques include cell fusion and the use of recombinant DNA or gene splicing. In cell fusion, the tough outer membranes of sperm and egg cells are removed by enzymes. And then the fragile cells are mixed and combined with the aid of chemicals or viruses. The result may be the creation of a new life form from two species, a chimera. Recombinant DNA techniques transfer a specific genetic activity from one organism to the next through the use of bacterial plasmids. Small circular pieces of DNA lying outside the main bacterial chromosome. And enzymes, such as restriction endonucleases, which cut the DNA strands, reverse transcriptase, which makes a DNA strand from an RNA strand, DNA ligase, which joins DNA strands together, and Taq polymerase, which can make a double-strand DNA molecule from a single-strand primer molecule. The recombinant DNA process begins with the isolation and fragmentation of suitable DNA strands. After these fragments are combined with vectors, they are carried into bacterial cells, where the DNA fragments are spliced onto plasmid DNA that has been opened up. These hybrid plasmids are then mixed with host cells to form transformed cells. Since only some of the transformed cells will exhibit the desired characteristic or gene activity, the transformed cells are separated and grown individually in cultures. This methodology has been successful in producing large quantities of hormones, such as insulin, for the biotechnology industry. However, it is more difficult to transform animal and plant cells. Yet the technique exists to make plants resistant to diseases and to make animals grow larger. Because genetic engineering interferes with the processes of heredity and can alter the genetic structure of our own species, there is much concern over the ethical ramifications of such power, as well as the possible health and ecological consequences of the creation of these bacterial forms. Some applications of genetic engineering in various fields are agriculture, crops having larger yields, disease and drought resistancy, bacterial sprays to prevent crop damage from freezing temperatures, and livestock improvement through changes in animal traits. Industry, use of bacteria to convert old newspaper and wood chips into sugar. Oil and toxin-absorbing bacteria for oil spill or toxic waste cleanups, and yeasts to accelerate wine fermentation. Medicine, alteration of human genes to eliminate disease, experimental stage. Faster and more economical production of vital human substances to alleviate deficiency and disease symptoms, but not to cure them. Substances include insulin, interferon, cancer therapy, vitamins, human growth hormone ADA, antibodies, vaccines, and antibiotics. Research, modification of gene structure in medical research, especially cancer research. Food processing, rennin, enzyme, in cheese aging.
Do viruses contain both DNA and RNA?
Viruses have either DNA or RNA as their genomic material, whereas cells including bacteria have both.
How do cells become cancerous?
Cancer is caused by the unrestrained growth of cells. Cells that do not follow the rules of normal cell cycling may eventually become cancerous. This means that the cells reproduce more often than normal, creating tumors. Usually this happens over an extended period of time and begins with changes at the molecular level. There are more than 100 distinct types of cancer, each of which behaves in a specific fashion and responds to treatment differently.
What is the bottleneck effect?
The bottleneck effect is a term used to describe a population that has undergone Some kind of temporary restriction that has severely reduced its genetic diversity. A bottlenecking event could be an epidemic or natural disaster like fire or flood. It is hypothesized that the lack of genetic diversity among African cheetahs is due to some bottlenecking event in the species past.
What is wormwood?
Artemisia absinthium, known as wormwood, is a hardy, fragrant perennial that grows to heights of 2 to 4 feet. 6 to 1.2 m. Wormwood is native to Europe but has been widely naturalized in North America. Absinthe, a liquor, is distilled and flavored using this plant. Absinthe was banned in the United States in the early 1900s. Because it is considered habit-forming and hazardous to one's health.
What is the relationship between ancient plants and coal formation?
Coal, formed from ancient plant material, is organic. Most of the coal mined today was formed from prehistoric remains of primitive land plants, particularly those of the Carboniferous period, which occurred approximately 300 million years ago. 5 main groups of plants contributed to the formation of coal. The first 3 groups were all seedless, vascular plants, ferns, club mosses, and horsetails. The last 2 groups were the now extinct seed ferns and the primitive gymnosperms. Forests of these plant groups were in low-lying, swampy areas that periodically flooded. When these plants died, they decomposed. But as they were covered by water, they did not decompose completely. Over a period of time the decomposed plant material accumulated and consolidated. Layers of sediment formed over the plant material during each flood cycle. Heat and pressure built up in these accumulated layers and converted the plant material to coal. The various types of coal, lignite, bituminous, and anthracite, were formed as a result of the different temperatures and pressures to which the layers were exposed.
What is the role of the queen bee?
A honeybee society is made up of a single queen, a few male drones, and up to 80,000 female workers. The queen is the only female in the hive capable of reproduction. And she can lay as many as 1,000 eggs in a single day. While the queen's job is solely to lay eggs, the workers' jobs change over time. Young workers serve as nurses to the larvae that develop from the eggs. Older workers serve first as hive builders and then as foragers.
What are some causes of root damage?
Roots may be damaged by temperature extremes, drought, nematodes. and other soil microfauna such as springtails that nibble the succulent roots. When there is known damage to the roots, cutting back the shoot system helps. To re-establish the balance between the root system and the shoot system. Transplanting seedlings and other plants also damages roots, especially the root hairs.
Is aggressive behavior inherited?
Because it is difficult to characterize a phenotype for aggression. More research is needed before specific genes are linked to aggressive behavior.
Which part of the wheat plant, Triticum aestivum, is used to make flour?
Wheat is a monocotyledon grass whose fruit, the grain, or kernel, contains one seed. The endosperm and embryo of the wheat plant are surrounded by the pericarp, or fruit wall, and the remains of the seed coat. More than 80% of the volume of the wheat kernel is made up of the starchy endosperm. White flour is made by milling the starchy endosperm. Wheat bran constitutes approximately 14% of the wheat kernel and is found in the covering layers and the outermost layer of the kernel, called the alurone layer. Wheat germ is the embryo of the wheat plant and represents approximately 3% of the wheat kernel. Athoff, there are nearly 24 species of wheat. The most important ones for commercial use are common wheat, Triticum aestivum, sometimes referred to as Triticum vulgar, and durum wheat, Triticum durum. The varieties of common wheat account for 90% of the wheat grown worldwide. Durum wheat accounts for 5 to 7% of the wheat grown. And all other species account for the remainder of the wheat grown.
How does the root system of a monocot differ from the root system of a dicot?
The root system of a monocot is a fibrous mass of roots providing broad exposure to soil, water and minerals. The root system of a dicot consists of one large taproot, with many small secondary lateral roots growing out of it.
What does the term umwelt refer to with regard to animal behavior?
Umwelt was first used by Jacob von Uexkell, 1864-1944, to describe the part of its environment that an animal can actually perceive with its sense organs and nervous system. Animals display this concept in the wild, but the same behavior can be seen with domestic animals. For example, if a stranger comes into your house and tries to pet your dog, this may be perceived as a threat within the dog's umwelt. The practical side of this is that you should be aware of your pet's umwelt when introducing it to new experiences and people.
What is high-pressure liquid chromatography?
High-pressure liquid chromatography, HPLC, was developed in the mid-1970s. It differs from other chromatographic techniques in that the liquid is pumped through a short packed column under pressure, instead of relying only on gravity.
How is nuclear waste stored and regulated?
Nuclear wastes consist either of fission products formed from atom splitting of uranium, cesium, strontium, or krypton, or from transuranic elements formed when uranium atoms absorb free neutrons. Wastes from transuranic elements are less radioactive than fission products. However, these elements remain radioactive far longer than fission products. Transuranic wastes include irradiated fuel, spent fuel, in the form of 12 feet 4 meters long rods. High-level radioactive waste in the form of liquid or sludge, and low-level waste. Non-transuranic or legally high-level, in the form of reactor hardware, piping, toxic resins. Water from fuel pools, and other items that have become contaminated with radioactivity. Currently, most spent nuclear fuel in the United States is safely stored in specially designed pools at individual reactor sites around the country. If pool capacity is reached, licensees may move toward use of above-ground dry storage casks. The 3 low-level radioactive waste disposal sites are Barnwell, South Carolina, Hanford, Washington, and EnviroCare, Utah. Each site accepts low-level radioactive waste from specific regions of the country. But only EnviroCare uses above-ground storage. Most high-level nuclear waste has been stored in double-walled stainless steel tanks surrounded by 3 feet 1 meter of concrete. The current best storage method, developed by the French in 1978, is to incorporate the waste into a special molten glass mixture. Then enclose it in a steel container and bury it in a special pit. The Nuclear Waste Policy Act of 1982, as amended in 1987, specified that high-level radioactive waste would be disposed of underground in a deep geologic repository. Yucca Mountain, Nevada, was chosen as the single site to be developed for disposal of high-level radioactive waste. On July 23, 2002, President George W. Bush signed House Joint Resolution 87, allowing the Department of Energy to establish a repository in Yucca Mountain to safely store nuclear waste. However, some scientists still expressed concerns about the estimates of how long it would take for rainwater and snow to infiltrate the mountain and corrode the containers.
What is a variable?
A variable is something that is changed or altered in an experiment. For example, to determine the effect of light on plant growth. Growing one plant in a sunny window and one in a dark closet will provide evidence as to the effect of light on plant growth.
Among the invertebrates, which are the most intelligent?
Most specialists agree that the cephalopods, the octopi, squids, and nautilus are the most intelligent invertebrates. These animals can make associations among stimuli and have been used as models for studying learning and memory. Octopi can be trained to perform many tasks, including distinguishing between objects and opening jars to obtain food.
How do conservationists predict whether a species will become extinct?
Conservation biologists use population viability analysis, PVA, a relatively new method, to predict the viability of a species in a particular habitat. Computer modeling generates PVAs using life history data, genetic variability, and a population's response to environmental conditions, especially disturbances, to predict viability of a species.
Are turtles endangered?
Worldwide turtle populations have declined due to several reasons, including habitat destruction, exploitation of species by humans for their eggs, leather, and meat, and their becoming accidentally caught in the nets of fishermen. In particular danger are sea turtles, such as Kemp's Ridley sea turtle, Lepidoscelis kempii, which is believed to have a population of only a few hundred. Other threatened species include the Central American river turtle, Dermatemes mawii, the green sea turtle, Chelonia midas, the leatherback sea turtle, Geochelone inophora, the desert tortoise, Gopherus agassizii, and the Galapagos tortoise, Geochelone elephantopus. Source: U.S.
Is coffee native to Colombia and Brazil?
Although premium coffee is today grown in the mountains of Central and South America, the coffee tree, Coffea arabica, is native to Ethiopia. Coffee was widely used in the Arab world before its introduction to European society in the 17th century. North and South American coffee plantations were started in the 18th century.
How are animals classified?
Animals belong to the kingdom Animalia. Most biologists divide the kingdom into two subkingdoms: 1. Parazoa. From the Greek para, meaning alongside, and zoa, meaning animal, and 2. Eumetazoa. From the Greek eu, meaning true, meta, meaning later, and zoa, meaning animal. The only existing animals classified as parazoa are the sponges, phylum Porifera. Sponges are very different from other animals and function much like colonial unicellular protozoa even though they are multicellular. Cells of sponges can be versatile and change form and function and are not organized into tissues and organs. They also lack symmetry. All other animals have true tissues, are symmetrical, and are classified as eumetozoa.
Can organisms with more than two sets of chromosomes reproduce?
Organisms with an odd number of chromosome sets cannot produce gametes with a balanced assortment of chromosomes. Polyploidy is very common in plants, with 30 to 70% of today's angiosperms thought to be polyploid. When a tetraploid, 4N, plant tries to breed with a diploid, 2N, plant, triploid offspring are formed. An example of this is the seedless grape.
How many flowers need to be tapped for bees to gather enough nectar to produce 1 pound of honey?
Bees must gather 4 pounds 18 kilograms of nectar, which requires the bees to tap about 2 million flowers. In order to produce 1 pound, 454 g, of honey. The honey is gathered by worker bees, whose lifespan is 3 to 6 weeks. Long enough to collect about a teaspoon of nectar.
How has DNA become commercialized?
DNA has been extensively commercialized in its applications to plant biotechnology. Genetically modified organisms, gene therapy, gene patents, and applications to forensic science. DNA jewelry, artwork, and apparel can be purchased. Music CDs have been created based on DNA sequences.
How can animal thought processes be studied?
Determining how an animal thinks will involve scanning the brain of the animal while performing a cognitive task. Data would then be compared with humans. Although human functional MRI experiments are becoming common, such experiments on other animals are still quite rare.
What are the 3 types of photoreceptors among invertebrates?
There are 3 different types of eyes, represented by different types of photoreceptors, among invertebrates. They are, 1, eye cup, 2, compound eye, and 3, single lens eye. The eye cup is a cluster of photoreceptor cells that partially shield adjacent photoreceptor cells. The compound eye consists of many tiny light detectors, photoreceptors. Crayfish, crabs, and nearly all insects have compound eyes. Single lens eyes, found in cephalopods such as squids and octopi, are similar to cameras. They have a small opening, the pupil, through which light enters.
Why are some animals so brightly colored while others are so dull?
There are two general functions of bright coloration. The individual with such coloration is trying to advertise either to members of its own species, its conspecifics, or to those of other species. Within species, communication revolves around reproductive behavior. Male red-winged blackbirds, for example, use their brightly colored shoulder patches to advertise their territory ownership to potential mates and rivals. An experiment with zebra finches found that females were more likely to choose males as mates if the identification bands on their legs were red rather than some other color. Between species, communication is usually threatening. Warning, or aposematic, coloration is a method used by animals with stings or poison to circumvent attacks. For example, stinging insects like wasps and bees use similar color patterns of yellow and black to advertise their arsenal. Poison dart frogs, family Dendrobatidae, also use bright coloration in this way. Dull or camouflage coloration, as in a flounder, is an alternative strategy. By hiding, these species hope to avoid predation.
What are RFLPs?
RFLPs are restriction fragment length polymorphisms, which are variations in the short base sequences where restriction enzymes can cut DNA. By cutting two different DNA molecules with the same restriction enzyme, scientists can compare the lengths of the fragments. Two identical molecules will have identical fragments, while two similar molecules may be largely alike, with perhaps a few differences in fragment size. These differences in restriction fragment lengths are called polymorphisms and are used in all types of DNA typing.
Who first demonstrated that blood circulates?
William Harvey, 1578 to 1657, was the first person to demonstrate that blood circulates in the bodies of humans and other animals. Harvey's hypothesis was that the heart is a pump for the circulatory system, with blood flowing in a closed circuit. Harvey conducted his research on live organisms as well as dissection of dead organisms to demonstrate that when the heart pumps, blood flows into the aorta. He observed that when an artery is slit, all the blood in the system empties. Finally, Harvey demonstrated that the valves in the veins serve to return blood to the heart.
What accounts for the different colors of bird feathers?
The vivid color of feathers is of two kinds, 1. Pigmentary, and 2. Structural. Red, orange and yellow feathers are colored by pigments called lipochromes deposited in the feather barbules as they are formed. Black, brown, and gray colors are from another pigment, melanin. Blue feathers depend not on pigment but on scattering of shorter wavelengths of light by particles within the feather. These are structural feathers. Green colors are almost always a combination of yellow pigment and blue feather structure. Another kind of structural color is the beautiful iridescent color of many birds, which ranges from red, orange, copper, and gold to green, blue, and violet. Iridescent color is based on interference that causes light waves to reinforce, weaken, or eliminate each other. Iridescent colors may change with the angle of view.
What technique may be used to determine the presence and concentration of various chemicals in a solution?
Spectrophotometry is based on the principle that every different atom, molecule, or chemical bond absorbs a unique pattern of wavelengths of light. Using a spectrophotometer, scientists are able to measure the amount of light that is absorbed or transmitted by molecules in solution. This value of this measurement allows scientists to identify the chemical
What are the upper and lower shell of a turtle called?
The turtle, order Testudinas, uses its shell as a protective device. The upper shell is called the dorsal carapace and the lower shell is called the ventral plastron. The shell sections are referred to as the scuettes. The carapace and the plastron are joined at the sides.
What is a goiter?
A goiter is an enlargement of the thyroid gland caused by hypothyroidism, too little thyroxine. An insufficient dietary intake of iodine is a common cause of goiter.
How does a homing pigeon find its way home?
Scientists currently have two hypotheses to explain the homing flight of pigeons. Neither has been proved to the satisfaction of all the experts. The first hypothesis involves an odor map. This theory proposes that young pigeons learn how to return to their original point of departure by smelling different odors that reach their home in winds from varying directions. They would, for example, learn that a certain odor is carried on winds blowing from the east. If a pigeon were transported eastward, the odor would tell it to fly westward to return home. The second hypothesis proposes that a bird may be able to extract its home's latitude and longitude from the Earth's magnetic field. It may be proven in the future that neither theory explains the pigeon's navigational abilities, or that some synthesis of the two theories is the actual mechanism.
What is parsimony?
Parsimony is a method for choosing the simplest explanation among a variety of possible explanations for phenomena when decisive evidence is unavailable. In evolutionary terms, this means grouping organisms or their traits in hierarchies that minimize the occurrence of special events. For example, all mammals are descendants of a fur-bearing, milk-producing ancestor, even though some mammals, e.g., platypus, echidna, produce eggs instead of live young. The most parsimonious explanation is that live bearing is a secondary adaptation that occurred after mammals separated from the other groups of animals. The geographic distribution of species is known as biogeography. By observing similar organisms and their adaptations to different habitats, researchers can ask and then answer important questions about evolutionary change. In fact, the diversity of habitats visited by Darwin on his voyages with the HMS Beagle helped him to solidify his ideas on evolution.
Which mycotoxin is found in certain agricultural products and is believed to be carcinogenic to humans?
The fungus Aspergillus flavus produces toxic aflatoxins. This mold is found in soil, decaying vegetation, grains, cereals, hay, corn, peanuts, sweet potatoes, rice, and animal feed. It is believed that products contaminated with aflatoxins are carcinogenic to humans.
What are the ideal conditions for fungi to attack wood?
Fungi tend to attack woods when temperatures range between 50 degrees and 90 degrees Fahrenheit, 10 degrees and 32 degrees Celsius. Wood needs to be moist for fungi to grow. The most serious decay occurs when the moisture content of the wood is approximately 30%. Wood with a moisture content of less than 20% will usually not decay, and any infection will not progress. Wood that is too wet will not decay because the excess moisture does not allow fungi sufficient access to air, thus impeding their growth.
Who eventually showed that plant cells were totipotent?
In 1958, Frederick Campion Stewart, 1904 to 1993, a botanist at Cornell University, successfully regenerated an entire carrot plant from a tiny piece of phloem. Small pieces of tissue from carrots were grown in a nutrient broth. Cells that broke free from the fragments dedifferentiated, meaning that they reverted to unspecialized cells. However, as these unspecialized cells grew, they divided and redifferentiated back into specialized cell types. Eventually, cell division and redifferentiation produced entire new plants. Each unspecialized cell from the nutrient broth expressed its genetic potential to make all the other cell types in a plant.
Which woods are recommended for construction when the wood will be constantly exposed to moisture?
Decay-resistant woods such as redwood and cedar or wood treated with preservatives are recommended for use in construction when the wood will be exposed to moisture. The most effective and least toxic preservative is chromated copper arsenate, CCA. Lumber treated with CCA has a characteristic light green stain.
How do sperm work?
Cells called spermatogonium become sperm through the process of meiosis. A mature sperm has only half the DNA required for a functional cell, so it can't survive on its own. However, they do have flagella and mitochondria that power their journey through the reproductive tract in search of an egg. Human sperm typically die within 48 hours if they have not fertilized an egg.
What is unusual about the natural distribution of lichens?
Lichens are widespread because they are able to live and grow in some of the harshest environments on Earth. They occur from arid desert regions to the Arctic. They grow on bare soil, tree trunks, rocks, fence posts, and alpine peaks all over the world. Some lichens are so tiny that they are almost invisible to the naked eye. Others, like reindeer mosses, may cover acres of land with ankle-deep growth. One species of the genus Verrucaria grows underwater as a submerged marine lichen. Lichens are often the first inhabitants of newly exposed rocky areas. In Antarctica there are more than 350 species of lichens but only 2 species of plants.